Hello and welcome to plant reproduction. I hope that quiz went well. Good luck learning plant reproduction stuff. Remember to work on your IAs. So diving right into plant reproduction, all about flowers. So flowers are the reproductive structures of plants. And when those flowers start to grow, when the plant begins to get those guys to bloom, it's all based on gene expression. So flowering involves a change in the gene expression in the shoot apex. Remember that is the tippy top of where the plant is growing. A couple different kinds of growth in plants, vegetative structures like roots and stems and leaves. This has indeterminate growth. Remember that means that it just keeps on growing, whereas our reproductive structures, those flowers, do have a determinate growth. They stop, they get to a certain size and they're done. That is unusual, less common for those plants. So cell signaling is going to control when those flowers pop up, and the signal is almost always based on light. And so we're going to talk about light as being that signal. But what's kind of quirky is that it's actually not how much light there is, but how much dark there is. What is the length of time of the absence of light? And the basic idea is that we've got this gene, flowering time gene, activated by these phytochrome pigments that are light will change their shape and make them an active form and inactive form. And then that gene will transcribe messenger RNA, which makes a protein, which then binds to cells in that apical meristem, turning on the genes that make flowers. So that's the goal, is to activate the gene, to make the messenger RNA, to make the protein, to activate those other genes, which will cause those flowers to grow. Good. So I don't think you need that vocab review. If you do, Cognity is a great place to look up those words. So talking about that protein, this phytochrome plant pigment that sets off this cascade of events, is based on the idea of photoperiodism, that we have repeating lengths of light and dark. So this phytochrome we refer to as PR in one version and PFR in the other. So PR, phytochrome red, absorbs light. It doesn't have to be specifically red light, like white light includes that red light, so it's going to be always there in the sunshine. It's not like it has to wait for a red light bulb. Anyhow, so that phytochrome red is going to be converted into phytochrome far red in the light. And then in the dark, far red will turn back into plain old red. So phytochrome red can turn into phytochrome far red in the light. Phytochrome far red turns into phytochrome red in the dark. Red is not active. It doesn't bind to any receptors. It doesn't do anything. It just sits there until it gets until it absorbs some energy from the sun. Far red, however, is active, but what's weird is that it can be both a promoter and an inhibitor. So far red, which we make in lots of light, is going to promote or activate the flowers in a long day plant, whereas it's going to inhibit, stop the production of flowers in a short day plant. And you can guess that long day plants are those that like longer days in the summer, whereas shorter days are going to be our early spring or fall plants that prefer a little bit less light. I'm sorry, my writing tablet here is a little fussy. Anyhow, so what's interesting again about that short day, long day, these guys like lots of sunshine, we say, and these guys prefer a shorter day. It's actually not about the length of the day, it's about the length of the night. And so if I have a short night and a long day, no flowers in my short day plant. If I have a long night and a short day, happy flowers. But here's weird. So I've got my long night with a small interruption of light. I've got my short day, my short day, my short day. Plant should be happy, but there's no flower because it's actually not about how much light there is. It's about how much dark there is. So a long day plant actually wants a short day plant wants a long night. Short day is better described as long night. And we can flip that for our long day plants. It's really not about wanting lots and lots of sunshine. It's about wanting a short chunk of darkness. So here, lots of sunshine, happy flower. Little bit of sunshine, no flower. Little bit of sunshine, but also we interrupted that night and we end up with flowers because he thinks he has a short night. So long day, short day, not the greatest descriptors. It really should be long night, short night. This is just showing another picture. So this is our phytochrome red being activated by the sunshine into phytochrome far red. This is the active version. This will be either our inhibitor or our promoter, depending on whether we're looking at a short day plant or a long day plant. So what do these flowers even look like once we get them bloomed? So we have petals, of course. The job of these guys is to attract pollinators. We have the stamen. This is the boy part of the plant. Filament anther. Pollen is on the anther. We have the stigma, which is sticky. Pollen is going to get stuck to the sticky stigma, which is attached to the ovary and the ovules. This is the female part of the plant. Our little eggs are here. Our little sperm are here. The job of the sepals, these are just little leaves to protect the flower while it's developing, or if it's a flower that closes at certain times of day or night, it protects that flower then too. I think we went through all these functions. So petal is all about attracting pollinators, sepal is for protection. This anther filament is the boy part of the plant, makes pollen. There's sperm and pollen. Stigma is sticky. It collects the pollen, sends it down into the ovary. In the ovary are ovules. In the ovules there are eggs, where fertilization is going to occur. So pollination, again, is that transfer of pollen from an anther to that sticky stigma. And plants are pretty cool, and they can self-pollinate. They can fertilize themselves, or cross-pollination. That pollen can go from one plant to a different plant. The benefit of cross-pollination, of course, is going to be increased genetic variation, which is good for a population to survive in a changing environment. But if that's not possible, self-pollination is a nice backup because at least you have some babies, even if they're not genetically diverse. Fertilization, pollen lands on the stigma. The sperm is going to travel through this pollen tube down through, we go from stigma to style, through the ovary into the ovules where those eggs are. How do we get that pollen onto the stigma? It is symbiosis. And this is just a quick review of symbiosis. So I've got my alligator not actually eating this plover bird, but instead the bird is cleaning his teeth. So this is an example of mutualism. They both have a benefit. My ugly mosquito here, this is parasitism. So he's feeding off the host without killing the host. The evil ladybug eating the aphid. This is pure predation, that fierce carnivore, the ladybug. And down here we have what are known as cattle egrets. So these birds just sit around the feet of cattle. And as they walk around the fields, the cattle disrupt bugs. The bugs hop. It's easier for the birds to see the bugs. So the birds get a benefit. The cow makes it easier for the birds to find a snack. The cow doesn't really get a benefit, but neither is he hurt. And so this is known as commensalism. One has a benefit and the other is neither helped nor harmed. Pollination, of course, is mutualism. Both organisms are getting a benefit. The birds are getting some snacks. This pretty little bat is getting some snacks, and then these plants are all getting pollinated. I'm probably gonna have to pause here. My video can only go 10 minutes, so I'm gonna pause here and then wrap up these last couple slides in another video. Good.